Good morning, and I'd like to thank the organizers for inviting me to speak this morning. And in terms of the different types of technology, I'll be talking about imaging technology and some of the types of imaging technology and mostly the analysis that we've been doing at Amgen in the last couple of years. And so I'll be talking about single-cell multiparameter image analysis. And so one of the things that we've been doing is just capitalizing on actually imaging single cells and being able to sort of leverage the information we can get from single cells in our screening. So the first thing that I want to talk about is a system called the FUCCI cells. And so this is a system that we're using mostly for some of our oncology programs, but FUCCI stands for fluorescent ubiquitination-based cell cycle indicators. And what this is it's basically two fluorescent proteins that are fused to genes that are associated with nuclear proteins. And so one is CDT1 and the other one is geminate. And these two proteins are sort of expressed reciprocally in the cell cycle. So what you would see is that you would have the expression of the red fluorescent protein at the beginning of G1, and it becomes ubiquitinated and over at this point at the beginning of S phase, and the green fluorescent protein is expressed at the beginning of S phase and is degraded at the beginning of G1. So you have this ability in live cells to be able to see the cell cycle by looking at the colors of the green and the red fluorescent protein. And so what this looks like is these are FUCCI cells that have the green and the red fluorescent protein. And so what you can see here is that the cells that are basically green would be associated with the G2M phase. The ones that are red, this is more or less red, associated with G1. And these cells are synchronized so that when these are going, what you'll see is that they actually change colors based on their cell cycle. So one of the things that's nice about this is that you could actually image these live and you could track these, kinetically over time, and they don't need to be fixed. So one of our first experiments, we were just looking at the changes, treatment with Taxol. So Taxol is a microtubule stabilizer. And so what we've done is we've, plated HeLa cells and we just used increasing concentrations of Taxol. And we've imaged these and we've also stained the cells with Hertz. And so we imaged these on the Opera. And so we're imaging the Hertz channel, the green fluorescent protein, and the red fluorescent protein. And it's a fairly simple analysis. We're just we're segmenting the nuclei and looking at the red fluorescent and the green fluorescent. And so what you see here is the DMSO control. And so you see a fairly nice combination of cells. Most of them are more in the G1 phase. And then as we have increasing concentrations of Taxol, you can see this is a montage of increasing Taxol concentration, and you can see that the cells become more green. So one of the things that we've been able to do, again, to take advantage of instead of just sort of imaging the plate, what we've been able to do is you look at each cell. And so what this is, for those who are more used to looking at FACS types of plots, this is each of these spots. This is from a single time point, and each of these spots represents a single cell. And so when you look at the FUCCI cells, it sort of forms this horseshoe type of shape. And so in this case, you have increasing green fluorescence protein in this direction. You have increasing red fluorescence in that direction. So one of the things that we could do is we could take the projected intensities in this direction on the x-axis. And this shows a fairly clear bimodal sort of distribution of cells. And we could use MATLAB and we could use some other proteins to be able to automatically fit that curve and figure out the separation between the G1 phase and the G2M and S phase. And again, the same way, we could take this population of cells that is over here, and you could be able to, again, separate this out to define the difference between the G2M and the S phase. And so for those who look at things in flow, this is the same experiment that's been done on a flow cytometer, and you can see that you get also the same type of distribution and the same type of segmentation and very close, sort of percentages in both systems. So as you look at something like a dose-response curve, what you expect to see is with increasing, Taxol concentrations, what you would do is you get a higher amount of cells that are in G2M, and you get a lower number that are in the G1 phase. And the S phase is fairly constant. Another experiment, just looking again at sort of the kinetics of this, is that we've taken we've done a double thymidine block, and again, these are HeLa cells, and we've treated the cells with thymidine. And this blocks the cells in S phase. And so again, we've stained with Hertz, we've imaged on the opera, and again, just basically doing segmentation on the nucleus and looking at the intensities of the green fluorescence, red fluorescence. And what you can see here is this, again, is the population at 0 hours, at 6, and at 12. And what we've done, again, is just using the automatic segmentation. And you can see what you'd expect is blocking something at S phase. What you would expect is a fairly large population of cells that are in this sort of quadrant up here that's associated with S phase. At 6 hours, you're progressing through the cell cycle, going into G2M, and you'll see that population down here. And at 12 hours, most of the cells are in sort of G1 phase. So this is a nice way that you could actually, again, so without actually fixing the cells, you could follow this population going through out of the thymidine block into the different stages of the cell cycle. And one of the things that, at least as a fairly inexpensive way of looking at things in the cell cycle, you could actually just stain the cells with Hertz. And a lot of times people do that. So when you're looking at this particular graph, this is looking at the integrated intensity from the Hertz channel. And a lot of times you could just basically model the cell cycle looking at the integrated intensity of Hertz. So this first peak is associated with the 2N peak, and this is the 4N peak over here. And if you use different cell cycle modeling, what you could basically do is you could look at these peaks and do some curve fitting. You could come up with sort of the percentage of populations of the cells in the different phases of the cell cycle. And what we can do, again, with the FUCCI cells is we could take the cells that are over here. You could basically do the same segmentation. And what we've done is we've taken the population of cells here and projected those cells onto this particular graph. And you can see, again, sort of the cells that are in 2N, 4N, and S phase over here. And it has a very nice correlation between sort of the Hertz staining and sort of a confirmation that the cells that you see in, that are coming from the HeLa model project very nicely onto, or the FUCCI project very nicely onto the Hertz integrated intensity. Another program that we've done to follow cell cycle is we've been using EDU, and EDU is a thymidine homolog. And so again, what you could do is you could tell the cells that are going to be in S phase based on the incorporation of this EDU compound. And so typically when you see the EDU graphs, again, this is looking at the single cells, what you would see is this is the integrated DNA intensity along the x-axis, and you're looking at EDU concentration or EDU intensity on the y-axis. And so typically what we could do is we could find the difference, sort of the minimum and the peaks here, figure out the, a cutoff for the EDU cutoffs. But what we've done here is I've taken the, a segmentation of a gating on the EDU channel and then projected that onto the FUCCI cells. And then again down here, projecting that onto the, if we look at the integrated DNA intensity, we can see how that projects onto that. So again, it's a very nice confirmation that these systems are all looking at the same things, as well as when you have the sort of the ability, if you look at the FUCCI gate, you could, again, this is the S phase in FUCCI that we've created the gates for. We could project that onto what we see in the EDU, and again down into the integrated DNA intensity. So what we've done is we've been able to do a lot of this automation in MATLAB, because basically you don't want to be sort of creating the gates yourself. You want to be able to automate all that process of finding the gates and creating the cutoffs there. And we've also used a tool called FCS Express. And this is a company that traditionally had been working in flow cytometry, and they've been sort of moving into using sort of that flow cytometry model as a way of looking at images. And so basically what we could do is we could move the single cell data from the opera or the operetta into FCS Express. You basically get the tables. You can get all the different types of, scatter plots, be able to, figure out dose response curves, looking at, again, sort of density plots. And the thing that's actually very unique about this program is it actually looks at single cells. So when we look at the single cells that are in these graphs, we actually can see those within these images. So if you happen to hover over these particular cells here, what you would do is you would see in this particular image, you would actually see the specific images that the actual cells that correlate with those data, the data points in these graphs. So the next thing is I just wanted to talk about that was fairly simple. We're basically just segmenting on nuclei, looking at intensity. What I wanted to do is to look at something that's more of a phenotypic, what I call a phenotypic screen, something that is much more complex than just looking at a single segmentation of the nuclei, which is very easy, and just looking at intensity. And so when we start to look at this, the process that we've gone through, basically we're doing image acquisition with the opera or with any type of imager. And then we're going through image processing, basically taking out the different features that we're looking for. So this would be the process of segmentation, looking at things like different morphometric measurements, looking at texture. Going the next step is doing some type of normalization and quality control. So this would be, we've been using again a tool called Gene Data Screener for this, and this is looking at things as you look at plate-to-plate variability, or being able to normalize all of your values in your plate. So using things like robust C to do that. The next part is to do some type of dimensionality reductions, being able to figure out which features as you start to get more and more features, which features do you actually follow and how do you actually follow those. And so we've been using different packages like JUMP or MATLAB, as well as a program called CatName-MyCells, which I'll talk a little bit more about later. And then going to hit visualization, actually trying to figure out how do we actually, after we've done all this number crunching, how do you actually visualize the different hits that you have, and how do you actually figure out ways to classify these in a meaningful way. So this particular screen, we are using a cell line called C3T10.5, and what this is a mouse mesenchymal stem cell line. And so this has the ability to go in at least reported to go in a couple of different directions. So you could have things that move into more of an osteogenic or dipogenic or myogenic. And so we were interested in the ability of proteins, soluble proteins, to change the direction of development of these particular cells. So with our treatment, what we've done is used a secreted protein cDNA library, and this has 1,400 clones and about 1,000 unique gene IDs. And we've been doing this either with direct transfection or doing a supernatant transfer. Again, imaging in the opera analysis would be with the package called Columbus or Acapella. So in terms of feature selection, this would be some of the, these are the same cells that have been treated with different proteins, soluble proteins, and they go in a lot of different directions. And so as you start to look at this, it's not just looking for the nuclei and saying, well, this is brighter, dimmer, red, or green. You're looking at a lot of different characteristics. And so in terms of segmentation, what we could do is we could look at things like we could segment into the nuclei, the cytoplasm, these different vesicles. These we've been staining with a Lipitox screen, and we're looking at mitochondria. So we're basically just looking at three different, fluorophores, which staining the nuclei with Lipitox screen and the mitotracker red. But we're able to get, do segmentation on each of those channels in each of those features. And so we could do things like we could look at the specific vesicles, we could look at the area, the shape, the intensities of each of these. I'll talk a little bit more about some of the things that are unique in terms of looking at texture and co-localization. So one of the things that we have is we have, potentially lipids, or we have mitochondria, and we could look at different types of texture features. And so this, one of the ways that is fairly easy to digest is they have a thing called saddles, edges, and ridges. And so these are just different types of, the software is able to produce a lot of different types of texture features. So this would be, say, the original image of here. And then you have things like you could look for spots or things that look like ridges or things that are bright or dark. And so it's looking for these different types of features. And so one of the things that happens, though, is in terms of as you start to look at these particular features, say if you're looking at mitochondria that are looking at tubulin, things that are very fine structures, what you want to do is you want to have a kernel size that's very small. If you're looking at larger objects, maybe vesicles, you're looking at things like ER, you might want to have kernel sizes or something that you're looking for something larger. So as you're automatically producing these different texture features, you're getting not just 9, but you might be getting about 90 texture features for each channel. So you start to develop quite a few different measurements. Another thing that we've used is called threshold adjacency statistics. And this is, so the previous texture measurements gave you things that, like a ridge or a hole. Those are pretty understandable. What this does is it just basically looks at, if you look at the center pixel and you say, well, how many white pixels are next to me? So you basically make a binary image. And what you can basically do again is change the kernel size. You can get a lot of different texture features and figure out a lot of things that are happening. And unfortunately, when you try to report this to somebody, you say, well, you have a texture feature of 5, with a kernel size of 2. It doesn't really mean a lot, as opposed to if you, gave them something that was more, easy to digest. But these are things that actually come up, as things that differentiate these cells. Another thing that we've been using a lot is co-localization. And so this is, what I have here is just this image here to talk about, these are the same images here that are thresholded on different intensities. And so one of the things that, often people use is just a co-localization method where you say I have an intensity in one channel and intensity in another channel, and if both things are above that intensity, they must be co-localized. In a lot of papers you see you have something red and something green and it looks yellow and you say, they're co-localized. But that's very dependent upon the scaling and the thresholds. And so we choose not to usually use those, that kind of qualitative method, but actually use quantitative methods of things like Pearson's correlation coefficients that are much more, they're sort of not influenced by these threshold cutoffs as much. So the next thing is we sort of move into the idea of dimensionality reduction. So when you actually, go through and find out a lot of these texture features and other types of measurements, you might be able to come up with 800 different measurements per well per, And then on top of that, you have maybe 1,000 cells per well. So you actually have quite a few measurements of each cell and all of these per well. And the question is, how do you actually handle all that information and what do you do with it? And so one of the things I originally thought that would be very nice is you could just do a scatter plot matrix and just sort of look at each variable and maybe something will pop out. You would say, obviously it's like these two variables, are the ones that are important, and I could differentiate between my positive and negative controls. But if you could picture a matrix that's 700 or 800 by 800 and trying to figure out which ones are really important, it's not very easy. And so one of the things that we've used is a software package called Cafe MyCell. And what this does is it uses a couple of different techniques. So you have, one of the things that it does is it basically asks you to get your positive and your negative controls and another, potentially something that's in between. And what you're able to do is, the first part that it's doing is you're taking your control samples and you're using something like, KS statistics or principal component analysis, and you're trying to figure out, well, which of those features of all these 700 features are really important? And then what it does is it then tries to, it basically reduces those factors, figures out which ones are important. And then it says, well, using those particular factors, which particular method of splitting can I use? So in this case, it's k-nearest neighbors, support vector machines, or neural networks, saying, of these, which of these particular machine learning techniques gives me the best answer based on those controls? And the thing that's nice about this program is it might be that you have 1,000 cells in your control wells. What it does is it randomly chooses cells within your control population. And so what it's trying to do is it's trying to take the user bias out of saying, this is obviously my control cell. And it basically lets, sort of the statistics sort of talk for itself and being able to separate out these different populations. And so then again, what you would do is after you find out sort of the classifiers and the best way of separating these, and you use this on your own data. So again, this is, what you would see is something over here. This is, a lot of different treatments. And then you have just, out of all of these, it's actually just pulled out about a half a dozen, particular features to use in that, to be able to use that within the principal component space. And you can see a very nice separation of your positive and negative controls. Another thing that we've used is this sort of a linear classifier. So in this case, what you're doing is this is again sort of, and I think we've moved away from this because what this does is it really sort of comes into a user bias where you go in and you could look at a population of cells and you say, well, these are my positive cells and these are my negative cells. And what this particular program, Columbus, can do is it can say, well, I'm finding out all the different, characteristics that make up that particular cell. And what I'll do is I'll go through and I'll look at the population and say that the things that are green, I've selected as being part of this population, and things that are red are that population. And one of the things with this is that you actually only get a couple of different populations. Maybe, with this it works pretty well up to about 3 populations. But one of the things that you have is, as we were doing our controls, this is using, again, sort of a different way of classifying. Instead of just saying, I can actually see my positives, I can see my negatives, and I'm selecting those, using a process called discriminant analysis. And what this does is, to me, it was a verification of our different types of segmenting and classification techniques. And so what we have, again, are, say, different conditions over here that we've, tested, different proteins that we've added to the cells. And what we have over here on the y-axis are the actual cells. So these are the actual populations. And then over on the x-axis, you have the predicted. So this is, I love the term for this, is it's a confusion, they call it a confusion matrix. But what you're doing is you're trying to say, of my population, the actual population of, say, A, what you have is using my different tools, I've been able to segment out and say most of these are A and some of them are B. They look pretty much the same by eye, but the different morphometric parameters we've been using can actually separate those out and start to differentiate between those two. And you can see the different populations, some that are a little more obvious than others. And it probably makes sense that as you actually look at these individual cells, that you don't have, one of the things is as we're using these protein libraries is you're not getting exactly uniform changes. The cells themselves are pretty different within these populations. And so that's sort of reflected, I think, in this confusion matrix over here. So another assumption that, people have when you look at, a lot of the cell-based population is you might think that you have a lot of the statistics people use are based on the assumption that you have a normal distribution within your population. And the idea is that you have a large enough number of cells and you should have a normal distribution. And then you use the appropriate statistics that you often feel I'm doing t-tests and I get sort of figure out this is different from that. But when you actually look at HCS data, what you see is populations of data that are very not normal. So this would be, on the left, what you have is, say, the measurement of nuclear roundness, and 1 being very round and, 0.1 being very unround. And what you can see is that in this population, it's very skewed towards roundness. So it would be skewed, I guess, to the right in being more round. In the graph on the right, what you see, so this would be in these charts, you have your basic histogram here, you have a box plot here showing that you have if we're looking at mito intensity, you have a huge number of outliers over here. And again, this is, shown in this called the normal quantile plot. So if you had a normal distribution, what you'd expect is to see your cells or your population going along that line. When you have things that are very not normal, you can see that they move and deviate off the line quite a bit. And this is showing that you have, this is reflecting this population over here, which is all your outliers. So the thing is that if you look at most of your HCS data, most of it is very not normal. So one of the things that we've been using is a Kolmogorov-Smirnov test, which is archaic statistics, which is easier for me to say. But what this is doing is it's using a non-parametric analysis that says, is one population different than another? And the thing that's nice about this is it doesn't assume that the distributions are normal. It's a non-parametric type of statistic. And so what you can do here is, again, you can, look at your 2 different populations. And what this is on your x-axis is whatever you're measuring. On the y-axis is this cumulative fraction plot. So this is, starting at 0, and this is your total population up here. And so you're looking at the distribution of your population and seeing the differences in those populations. So when we look at something like, these cells treated with TGF-beta, what you can see is that you have some parameters that don't really change at all and some that are very different. some that are, in this case, if you're looking at this one, this is some measure, and our treated population becomes very small, whereas the normal population untreated is fairly far over here. So one of the things that's nice about the KS statistics is that you could use different populations for your whatever you're comparing. So if you're saying you want to drive, finding something that looks like some particular treatment, you could use that as your control population and compare everything against that and see if things are very similar or very different. So what we have is the ability to produce, thousands of data points per well and per cell. And so the question is, how do you actually, again, sort of figure out which things do you want to look at? So here would be an example of hierarchical clustering. So we've, again, taken all those different single measurements, that from the single cells and created sort of a well-based averages based on those treatments are. And so on, say, this y-axis, what you have are the different compounds, and on the x-axis would be your different things that we've measured. And in terms of hierarchical clustering, what you could think of is you could take these populations, and they have basically two different ways of looking at it. One is that you could sort of clump everything together, and you split from the top, and you say this group of things is different than this, and you end up with a tree that says, I'm splitting. Or you could start from the bottom, and you say everything at the bottom is different, and you're going to look for things that are similar, and you group those together. And this is actually, fairly informative. So you can, see in terms of some of these particular things that we're measuring, a lot of them are showing the same sort of characteristics. Here you can see these are all sort of measuring the same type of things. So it might be that this is a group of texture features of the mitochondria. And they all basically don't, one measure is basically as good as another because they're all telling you the same piece of information. Another thing that we've used is, and I think this is, to me, a little more informative, is the k-means clustering, where you basically have to go in and say, well, how many different clusters, how many different populations of things have I produced by my treatment? And what we've done here is we've selected, in this case, 11 different clusters. And so what this does is it starts off initially saying I'm taking sort of 11 different things that look fairly random and far apart, and starts those as the center of a cluster. And what it tries to do is it tries to sort of, put the different, what it'll do is it'll try to minimize the differences within these clusters, and once you'll end up with different populations that are similar to each other. And so this is very nice because what it ends up is that you have all these different measures, and it says these things produce things that are similar to each other. And the nice thing is that as we've been able to sort of move and, as we've been sort of reporting this back to the group that we've been doing the screen for, what they can do is they can sort of see these things that are clustered together, and they make sense as they're clustered together. And another way that we've actually been able to look at this is called parallel line charts. And again, what you have is each of these lines is a different treatment, and on the x-axis are all the different measurements. And so one of the things you could see with this is that these are some of the different controls, and you can see how the different controls group together. But again, this is something that is, you could sort of go through each line and think, well, that's sort of interesting, and try to pick those out. But we've also used another measurement, which is called the Mahalanobis distance, which is basically taking all of those distances and saying, well, what is my, I have something, and then how different is what I'm measuring from that control? And so, so this is actually shown in an alphabetical order. So you have some things that are fairly, so the things that you could do is you could say, I'm driving for something that's very different than my controls, or I'm looking for something that's very similar to my controls. So you have the ability to sort of do that. So if you actually took that data and you put it on sort of a grouping where you'd have things that are very different from my controls to things that are very similar to my controls. So this gives you an ability to sort of, again, validate, are your controls working, and again, sort of lumping these together. And so the last part is, again, sort of using principal component analysis. And what this is doing, the thing that we've used this a lot for is that this, again, is, with principal components, what it's really asking is you have an initial set of variables. And these are all these different, it might be length, width, intensity. And what you're doing is you're taking those measurements and you're saying, in your first principal component, you're making a linear transform of that and saying, I'm taking the things that produce the biggest difference in my population, and that'll be my first principal component. And then the second principal component, what it's basically doing is saying what is orthogonal, or what is orthogonal to that particular line that describes the greatest distance or the greatest difference in the population that wasn't found in my first axis. So the way that this looks is that, these are the different principles. So if we had 40 different features, we would have, 40 different principal components. And what you would see is that these are the ones that are selected, and weighted the most in that first principal component. And as you go further along, you have fewer and fewer differences that your principal components can see. But what you can see here is this is just basically asking, how much of the variability within my population space is contained within that first principal component. And what this says is about, 37% of the variability is found in that first principal component. So you could usually use about the first three principal components, and you can see a very nice separation of your data. And so what we've done is we've been able to, again, this is these things in this principal component space, and the things that are, say, way over here would be something like, these are these cells that have been treated with TGF-beta or Wnt, and these have, you can see that they have a huge amount of, sort of a large mitochondria, very little, have a lipid reduction. If you, treat with BMP4, what you see is an increased adipogenesis. So you can see that, reduction in mitochondria, just, huge sort of lipid droplets. And, sort of something in between. So as we, and the thing is that those are our controls. We knew that those things are going to be really far out. But then the thing that's nice about viewing this in principal component space is you could actually see the things that are lumped together, sort of in these things that are not in this, one-dimensional axis, but the things in the second principal component sort of pull things off the axis. You can see things that are grouped together that way. So we then actually can use some of the different clustering methods and find different clusters within that principal component space. And again, that's been very promising. And so as we're doing that, sort of the next step is actually taking, again, we've treated these cells, and we're going to sort of go to the FUCCI and sort of go through and actually look at some of the sort of the expression of these particular proteins, and sort of look at some different, within each of these treatments, do you have sort of, are you turning on similar genes within these? Okay. And so anyway, I'd like to thank a lot of my colleagues at Amgen, Perkin-Elmer, De Novo, and Mac Biophotonics. And thank you for your time.